And Danielle's presentation this morning is identification and genetic mapping of novel sources of resistance to groundnut, rosette, and late leaf spot diseases in a collection of Arachis wild derivatives. So thank you very much, Danielle. It's now your time.
Hello, everyone. My name is Danielle Essandoh, and I'm presenting to you today on the identification and genetic mapping of novel sources of resistance to GRD and late leaf spot diseases in a collection of wild Arachis species. So groundnut is a very important crop here in Uganda. It's the second most important crop after common beans, and it's grown in the eastern and northern part of Uganda, and then for making it a crop of economic importance in Uganda. So despite the economic importance of the crop, its production is affected by late leaf spot, causing 60% yield losses annually, and groundnut rosette disease, which causes about 100% yield losses annually. Therefore, whenever novel lines are being introduced into the country, we need to know their responses to these two diseases and also their yield potential. These are some of the symptoms of the diseases. So several methods have actually been identified for the control of both late leaf spots and GRD, of which host plant resistance is the most efficient. The groundnut breeding program in Uganda has developed several lines that have late leaf spot and groundnut rosette in their background, such as the Serenat 2 and the Serenat 4. However, some of our lines are still succumbing to this high disease pressure, and they still have some, their low potential is actually their yield potential is actually somewhat low. So the problem also comes from the low genetic base of the crop, which therefore making it, which has few allelic combination for disease resistance. The cultivated groundnut was actually derived from two diploid species, spontaneous duplication and domestication, which has prevented it from sharing alleles with its wild parent, therefore making the genetic base of the crop low. However, this has been breached by the breeding program in Senegal, and they've developed interspecific lines which introduce wild alleles from 4 different species. We could actually use these lines as parents in our breeding programs. However, there's limited information on their responses to late leaf spot and GRD, and also their yield potential. So aside the disadvantages of conventional breeding approaches, phenotyping is also somewhat time-consuming and expensive. However, when we identify QTLs, which is the basis of marker-assisted selection, this will actually save time and expenses. Therefore, my objectives were to determine the response of interspecific lines of groundnut to groundnut rosette disease, late leaf spot, and determine their yield potential. Also to identify QTLs associated with the groundnut rosette disease and late leaf spot. And I hypothesized that the interspecific lines outperform our local checks for disease resistance and yield. And the QTLs associated with late leaf spot and groundnut rosette disease actually exist due to the wild introgressions. So this, for the first study, which was evaluation for disease resistance and yield, the lines consisted of the lines generally with the interspecific lines with 376, consisting of lines from A. valida, A. duranensis, A. ipaensis, A. duranensis, and A. batizocoi, A. duranensis. And we evaluated them with 3 local checks and the recurrent parent. So the lines were evaluated in 2 hotspots of late leaf spots and groundnut rosette disease in Uganda, which have been indicated with a star for 2 seasons using a 19/20 alpha lattice and 2 replications. So data was collected at 4 weeks, 8 weeks, and 12 weeks and at harvest, and also yield traits were taken. Percentage disease incidence was determined, and yield data collected included the number of days to flowering, seed yield, and pod yield. These were the scales that were used for on the field for evaluation. So for the results and discussion, the lines actually varied across the 3 locations. They were significantly different from each other, which is actually very important for breeders because we need diversity to aid us in selection. However, particularly important for me was the A. valida population, which was more variable in comparison to the other 2 populations for late leaf spots and groundnut rosette disease, which is actually very important in our breeding program. However, the the lines were actually skewed towards for late leaf spot. They were all, the median actually indicated they were susceptible, indicating that most of the lines were actually very susceptible. But for GRD resistance, you find that some of the lines were somewhat resistant. So this is just to show the yield comparison of our local checks with our interspecific lines. Our local checks were the standard checks for the Groundnut Rosette disease. However, you find that some of the lines, about 8 of them, actually outperformed our local checks for yield, an indication that we can actually introduce some of these novel alleles into the background of our local checks to improve their yield potential. It would also be helpful to elucidate the mechanism of resistance that actually occur in these lines. As Groundnut Rosette, the ancestor virus has been reported to reduce yield losses without showing any symptoms. Okay, this is also to compare our interspecific lines with our best local check, which was SERENA2. The red line is an indication of where our SERENA2 is, and the black line is actually where the breeding program will actually call it the threshold. So you find that several of the lines were below the threshold, an indication that we could use some of these lines to improve our local checks, and also commercialize and use some of these interspecific lines, which combine yield and disease resistance. So the second objective was to identify QTLs associated with resistance to late leaf spot and Groundnut Rosette disease. So for that objective, 133 interspecific lines from the A. batizocoi population was used in conjunction with the phenotyping data from study 2. All statistical analysis was done in R. The physical map in combination with the phenotypic data was then used for the QTL identification. So this is just the results for the heritability estimates that were obtained. We had moderate heritability estimates for the lines for late leaf spot and Groundnut Rosette disease, of which Groundnut Rosette disease PDI at 4 weeks and 8 weeks were quite low, indication that the lines were not very variable for Groundnut Rosette disease, as they had been I think they had been preliminary bred for yield. But you find that some of the genetic component, the heritability estimates were quite high for late leaf spot and late leaf spot at 12 weeks, and AUDPC is an indication that to get better results for evaluation, it should actually be done at 12 weeks, especially for this particular population. So for the linkage map, this is actually the linkage map we had, and we identified 4 QTLs for Groundnut Rosette disease, of which 3 had moderate phenotypic variation explained, which was greater than 10. These QTLs are novel QTLs that could actually be used for marker-assisted selection. Late leaf spots, we identified 2 QTLs for late leaf spot, which were minor. They were less than 10. However, from studies, we realized that late leaf spot is a quantitative trait which is controlled by both minor and major genes, and therefore these genes could actually be validated in another population, in another diverse population. So to determine the effect of the wild alleles on the trait of interest, for all the trait of interest, the wild alleles actually contributed to the lower trait values of all the traits, the QTLs we observed. This actually confirms the value of the diploid species as sources of resistance in groundnut breeding program. For Groundnut Rosette severity, you realize that the trait values were actually reduced. You can also, this actually indicates that there's some level of resistance in the cultivated background, which can actually be projected by using our wild species. So in conclusion, 17 lines from the A. valida population were identified, which were resistant to Groundnut Rosette disease, of which these 2 lines, B73284, B72522-8, were able to combine resistance into a high yielding background. The QTLs identified for late leaf spot and Groundnut Rosette disease actually provides additional tools for marker-assisted selection. the way forward and recommendation, the lines were variable, especially for yield, and therefore the entire population should be preserved. A crossing program should be initiated to actually derive our local interspecific lines, which have been bred specifically for groundnut rosette disease and late leaf spot. So I would like to acknowledge the following organizations who have played a significant role in this work, and also believe that my work contributes strongly to the UN Sustainable Developmental Goals. Thank you very much.
Thank you very much, Danielle, and a very nice presentation. We'll open the floor to questions now, if there are questions. And if you'll type them in the question and answer box, I will read them out to Danielle. Any questions at this point? One question I have, Danielle, is, and if you'll turn your video back on so we can see you. The question I have for you is with these lines that you have now, how long do you think it will be before there's possible to have a variety that's adopted by and used by growers in Uganda and in surrounding nations?
I think it will take about roughly, okay, we've already started evaluating them again. So hopefully in 2 to 3 years time, we should actually release some of these lines.
Okay. And are they being tested anywhere outside of the same lines being tested elsewhere in the world, or primarily with you in the Uganda area?
And primarily in Uganda.
Okay, thank you.